The next two speakers are Rosemary and Toomey and Kim Sullivan. We've known each other for a very long time. Dr. Toomey and I did a study, I think back in 1998-ish together. It's the beginning of that. And Kim, not too much longer after that. One of the beautiful things in science is being a scientist that's so passionate about the work is that you run into other folks that are passionate about the work. And it not only leads to lifelong collaborations, but also lifelong friendships. And so I'm very grateful and for that experience. A little annoyed that we don't get to do person-to-person meetings as much as we used to, because that was where a lot of that came from, and screens aren't the same. But still. So Kim and Rosemary are both neuroscientists and psychologists at the University of Boston. They've been engaged in this work. Kim, I think the earliest person in the whole field was you, because you started immediately after the Gulf with the Fort Devens cohort. But since then, you've done a tremendous body of work. She's been a leader, a driver of the field. One of her superpowers is getting people to work and play well together, and coordinates a big consortia of folks that bring all kinds of skills and bringing new people to the field with amazing skill sets. I would give Kim tremendous respect and thanks for her bringing so many really amazing scientists to the Gulf War illness field, and then creating whole systems that let them be effective, such as the BBRAIN, the biorepository and data repository that links samples and specimens and data to the researchers that need them to move the field forward. So that's been tremendously important. Dr. Toomey is also a research professor at Boston University and a neuroscientist, and she's also been involved in Gulf War illness from very early on, tapped onto her earlier work that looked at neuropsychological effects of military deployment, PTSD, and addiction disorders. She's been doing this work for quite a bit, including twin studies from the Vietnam era, and longitudinal studies on aging that's important from the Vietnam era veterans. She started working with Gulf War illness in 1998 with a National Health Survey, and then with the BBRAIN cooperative study that I was involved with. That's how she and I got to know each other. And then she started working with Kim in 2013 on the Gulf Wellness Consortia, and now BBRAIN and the new BBRAIN studies. So no further ado, I pass it to you.
Thank you, Nancy, and thank you for inviting us to come and talk today. I'm always very happy to talk with our Gulf War veterans and update you on what we're doing. I'm going to be talking about the BBRAIN biorepository that Nancy mentioned that I lead as the director and principal investigator. BBRAIN was started in direct response from veterans telling us over the years to work together to develop targeted markers, diagnostic markers, and treatments for Gulf War illness. And I'm going to give a brief overview of our repository and some of our key findings to date. And then I'm going to turn it over to my colleague, Dr. Rosemary Toomey, a very talented neuropsychologist, who's going to give you further update of our cognitive study outcomes. So the BBRAIN repository is an example of a resource that was developed specifically to encourage collaboration and speed the process of scientific discovery. But it also serves as an important source of sample validation, and that's equally important for the role of a biorepository. And the BBRAIN includes more than 10 study sites across the country who've shared existing samples and data for our studies, and that's been incredibly important working together. And in addition, we're also collecting new data at 2 study sites in Boston and Miami still. So if you're interested in participating in any of our study sites, please do reach out to us. This is a menu of samples and data that's available from our consortia, from all the hard work of the investigators who've been willing to share their samples and data, and all of you veterans who've been willing to let us collect all of these samples, whether it's blood samples, saliva, or we send you home with we call it your homework of sending us your stool and your urine samples. And as you heard some of the talks today, we're actually using some of those samples. So just so you know, all these things we're asking you that we're collecting, we're actually using and sharing for ongoing studies. We also have cognitive, exposure, and health symptom data that is being made available to researchers to speed up that time of actually being able to let them conduct their research, and translating a lot of times from our animal research into clinical research, because we can provide the samples and data that they need to then translate their research. So in this paper, we describe everything that's being collected as part of the BBRAIN and that we're sharing with the research community. To date, we've had nearly 50 requests for samples and data that we're in the process of sharing. And as I mentioned, we've shared some of those samples with many of the speakers you'll hear from today, whether it's Dr. Chatterjee or Dr. Klimas and others. So we're actually very, very happy to be able to share these samples and data and build on these results. And we're thankful to all of you who've been willing to come and do our studies, because that's the only way this works. So what are our key results or accomplishments to date? So some of them include the results of brain imaging studies, where we were actually the first to show inflammation in the brains of veterans with Gulf War illness by using PET imaging radio tracers of a protein called TSPO. And these are located on glial cells within the brain. And you heard from Dr. Shetty about microglia and astrocytes. Those are these glial cells that we were seeing activated. And you can see in the figure below, which shows this TSPO PET ligand has been activated in veterans with Gulf War illness compared to healthy controls in the red and yellow here. And we have now been funded for several new brain imaging studies of PET imaging and machine learning, where we're going to tease out which type of these glial cells are causing the brain inflammation. Why is that important? It's important because this will allow us to develop treatment development pipelines with strategic targets with already FDA-approved drugs. So we're not going to develop new drugs that are going to take a long time to try and use with our veterans. We're talking about FDA already approved drugs that actually target these particular types of glial cells. So we've also found in our results of ongoing imaging and biomarker studies very promising objective diagnostic tests potentially for Gulf War illness. So first, we looked at brain imaging markers of white matter glial changes in the brain, and we found a 94% accuracy in classifying veterans with Gulf War illness by that Kansas Gulf War illness criteria that you heard about from Dr. Steele. We also found that levels of the chemical messenger glutamate was significantly higher in the blood of veterans with Gulf War illness compared with healthy veterans. And again, I know some of you are aware of ongoing treatments for that. And collectively, these results suggest strong targets for Gulf War illness based on these central nervous system changes, and that a simple diagnostic test may be well within reach for Gulf War illness. And there are also FDA-approved drugs and supplements available to target these central nervous system targets as well. So from the BBRAIN study and combining data sets from several of our colleagues, we were able to combine enough data to really see power on some of our cognitive test outcomes as well. And we actually were able to combine data from nearly 500 Gulf War veterans. And our results showed decreased attention, executive functioning, learning, and memory in veterans with Gulf War illness compared to our healthy controls. And we also found that veterans exposed to pesticides and sarin nerve gas had lower executive system functioning on their cognitive outcomes. Those exposed to oil well fires had worse verbal scores. And veterans who took those pyridostigmine bromide anti-nerve gas pills had differences in attention and verbal memory scores. So we definitely had enough power to see these associations of exposures and cognitive outcomes. I know we had some questions about that earlier, about some of these exposures. We have several new studies that we're recruiting for veterans to participate in as well. And these include the PET neuroinflammation imaging study. So in this study, this is a follow-up to the prior study I was just telling you about, where we're studying different types of glial cells in the brains of Gulf War veterans to determine whether they're more active in people with Gulf War illness. And then we will compare which cell types are responsible for this inflammation in the brain and how the disease impacts the structure of the brain. And again, that will lead us towards treatment development. And if you're interested in potentially participating in that study, you can reach out to our colleagues at Mass General Hospital who are the driving force for these studies, our colleague, Dr. Marco Loja, and you can reach out to our group as well. We're also still recruiting for a study that we're calling the new BBRAIN study. And this is a study that's recruiting veterans with Gulf War illness and exclusionary medical conditions that might have had you be excluded from other prior studies. So this non-exclusionary wave, or the new BBRAIN study wave, will give more veterans a chance to participate in research studies who may not have been able to in the past. And in fact, for this new BBRAIN study, we're enrolling 200 Gulf War veterans who meet the Gulf War illness criteria, that Kansas criteria, but also have a previous would have been a previous exclusionary medical condition. So things like diabetes or stroke or cancer or heart disease, things like seizure disorders, multiple sclerosis, Parkinson's disease, um, multiple different types of conditions. So if you have some of these disorders, maybe you've not been able to participate in other prior studies, please do reach out to us. And the goal of this study is to expand on the BBRAIN cohort and to gain insight on whether or not these conditions are related to more severe cases of Gulf War illness, or if Gulf War illness develops into some of these disorders over time as our veterans are aging. And we've done a series of research showing that we think our veterans may be having some accelerated aging. Here is our website to contact us. It has information about our studies and about BBRAIN, our email, our telephone number. There's a very simple, brief screening instrument if you'd like to go on there and get information and have us send information to you. We'd be happy to reach back out to you. And I just would like to say a very big thank you to all of our veterans who have participated in our studies over the years. None of our studies would be successful without veterans like yourself who've been willing to participate and who've had the patience to help us to get to the stage where we're at, where we think we do understand what is happening in terms of the cause of Gulf War illness, and we are getting really close to developing very, very targeted treatments for the illness. And with that, I'm going to pass it on to my colleague, Dr. Rosemary Toomey, who's going to tell you a little bit more about some of our cognitive outcomes, specifically subjective symptoms that our veterans have been telling us they're having with cognitive problems, and the objective cognitive tests we've been doing with them.
So I'm going to be talking, as Kim mentioned, about subjective cognitive decline and how it relates to biomarkers in Gulf War veterans. About a third, as you've heard already today, of the Gulf War veterans exhibit symptoms known as Gulf War illness, and neurocognitive deficits are some of the more distressing and well-documented symptoms. And as you heard Kim mention, veterans with Gulf War illness tend to have lower scores on neuropsychological tests of attention, executive functioning, and memory, even decades after the war. So, as Dr. Klimas mentioned, I've been working for 20 plus years on the Vietnam era twin study of aging, where we showed that mild cognitive impairment, which is thought to precede dementing illnesses, can be identified as early as one's 50s and validated by association with hippocampal atrophy and Alzheimer's disease polygenic risk scores. So the Gulf War veterans are now in their 50s, and I thought it would be interesting to look and see if we can start to identify signs of MCI in Gulf War veterans. And Dr. Chow did such a study and found that 17% of veterans met criteria for MCI, and that also found hippocampal atrophy in those veterans and differences in lateral ventricle volumes. So just to give you a definition, subjective cognitive decline reflects cognitive difficulties in everyday life, as opposed to cognitive difficulties that are assessed by objective cognitive tests. So it's thought that subjective cognitive decline may precede objective cognitive impairments and therefore be like an early warning sign of increased risk of developing neurodegenerative diseases. Initially, people focused just on subjective memory decline, but more recently it has been expanded to include other domains of cognition. So in the study that I'm going to be talking about, these are participants from the BBRAIN study, and I'm focusing on 221 from that study, of which 180 had Gulf War illness. Gulf War illness was defined by the Kansas symptom criteria, which you heard about earlier today, and for this particular sample, they did also have the exclusion criteria. So we use something called the Measurement of Everyday Cognition Scale, which is the acronym is ECog, and it's a self-report survey that asks, please rate your ability to perform certain everyday tasks now as it compared to your ability to do these same tasks 10 years ago. And there are subscales that focus on memory, language, visual, spatial abilities, planning, organization, and divided attention. So an example of an item might be, how good are you at recalling conversations a few days later compared to how good you were on doing that 10 years ago. And then we also have a number of cognitive tests, 8 different cognitive tests. We looked at cytokine levels in blood, and we looked at brain volumes via MRI. So what we did initially is compare cases and controls on demographic variables and the ECog. Then we looked at the association between the ECog and Kansas symptom domains. I think that's not listed here, but we did that, as well as the cognitive test variables, the brain volumes, and the cytokine levels. And we looked at correlations within cases and controls, and then did a statistical test to compare if they were larger in one sample or the other. So just in terms of demographics, the cases and controls didn't differ on sex, race, being Hispanic, Latino, employment, or branch of service. Overall combined sample was 85% male, about 80% white, 12.8% Hispanic, Latino, and 66% married. Cases were actually significantly younger than controls by a couple of years. They were more likely to be married or living with a significant other, and they completed significantly fewer mean years of education, although both groups were highly educated. So was not the focus of it, but just to inform you, there was significantly more depression and PTSD symptoms in cases compared to controls. And here are the case versus control comparisons on the different ECog subscales and then the overall. And these are means. And so you see that this was significantly different, significantly more symptom reporting in cases. And of note, that was the case when you controlled for depressive symptoms, and also when you controlled for PTSD symptoms. In the PTSD and depressive symptoms tend to be very highly correlated, and in the field of subjective cognitive decline research, people focus a lot on depressive symptoms. So if I talk about controls after this slide, I'll emphasize the depressive ones. Here are correlations with Kansas symptom domains in cases versus controls, and then the final column tests the comparison. So this is now the mean total ECog, not the subscales. So you see it was quite correlated with all the Kansas symptom domains in cases with neurological and gastro symptoms and controls, and the correlations were significantly higher in cases compared to controls on fatigue and pain. And both groups had a high correlation with neurological symptoms. So the next 3 tables are looking at the correlations of the total mean ECog with objective cognitive tests. So the first line here is a test of like your general cognitive ability. And it's important to establish that was not different between cases and controls. That makes it easier to say that the differences on specific tests were not related to general cognitive ability differences. So you see here a number of significant correlations within cases between the ECog and several measures of visual attention. And here you see it with greater subjective cognitive decline associated with slower processing speed, more behavioral disinhibition, and worse cognitive flexibility and initial responses on a fluency test. And And on this slide, you see more subjective decline reporting associated with worse memory performance, both visual and verbal memory. Here you see the associations with the cytokines, which were really quite interesting to me, and I need to schedule a meeting with Dr. Klimas to further discuss the interpretation of these. But you see significant differences. You see it within cases relationship of the ECog to IL-10 and TNF-R1, and you see 3 different cytokines related to the ECog within controls. And when you're comparing these values, there's a difference between cases and controls in IL-1B and IL-12. And if you controlled for depression here, the cases are exactly the same pattern. And the controls, this ILB is not quite significant, so it takes away this significance of this comparison. And then we have the brain volumes. So no significant correlations in controls, and one with the subcortical limbic volume in cases, not significantly different between them, but this holds true whether you control for depression or not. So in conclusion, veterans with Gulf War illness reported more cognitive decline in the previous 10 years compared to Gulf War veteran controls, and this does not seem to be explained by mood symptoms. And in veterans with Gulf War illness, subjective cognitive decline is significantly associated with all Kansas symptom domains, and more so than in controls, in particular with fatigue and pain, although the largest correlation in both cases and controls was with neurological symptoms. And subjective cognitive decline is associated with cognitive test scores from various domains, like processing speed, visual attention, executive functioning, and memory. There was only one correlation that was significantly greater within cases, and that was on a measure of behavioral disinhibition. And we see, again, greater subjective cognitive decline associated with higher levels of anti-inflammatory IL-10 and pro-inflammatory TNF-R1, and differences in correlations on these 3 that were significant within controls. And see it associated with subcortical limbic area in those cases, and that area is associated with memory, thinking, and performing everyday tasks, which is what the subjective cognitive decline measures. So I think Gulf War veterans might be at greater risk for mild cognitive impairment, which is thought of as a precursor to dementia as they get older. And given that they're in their 50s now, it's important to think of monitoring them for subjective cognitive decline and MCI so that we can see who might be at risk for further cognitive decline, because there are a number of modifiable risk factors for dementia that might prevent or delay up to 40%. And education about these factors in those that are identified at high risk might help prevent further decline. And this slide just represents the number of colleagues, collaborators at 4 different locations that contributed to the study.
That was great. Thank you so very much. This was a really excellent session and a lot to be learned.